Okay, so hello everyone. I know this is the first time we're doing an online lecture, so to give you kind of a feel for what the online lectures are like, I'm just going to take one moment to get my pointer set up, which I always have to do at the very beginning. And let's see, we'll click pen, and let's see, we'll do, how about a blue pen today? We'll see how that works. So anyway, we're going to be doing the spinal cord lecture today. This is a pretty quick lecture, so my goal is to make this lecture about 45 minutes long, and then I'll stop this and record a little bit of the peripheral nervous system so we don't fall too far behind. So let's get started on the spinal cord. So I've already done the central nervous system, so you know that the central nervous system includes both the brain and the spinal cord. We've spent countless amount of time on the brain, and so we're going to spend a little bit of time on the spinal cord. The spinal cord is much less complex, as you can guess, than the brain is, but obviously, again, together, just as a reminder, both the brain and the spinal cord make up the central nervous system, so that's why this one's still kind of included with the central nervous system, even though we had to split it out for the next exam. So again, you can see here some of the basic functions for the spinal cord. Our spinal cord reflexes, which we'll talk about. Some reflex arcs go right through the spinal cord and never make it to the brain. And then we'll talk about integration of nerve impulses. So you'll see that we get some integration in the spinal cord where many sensory neurons will be coming in, and the summation of either those that are inhibiting and those that are exciting will be integrated actually in the spinal cord in some areas. And then remember that would determine those impulses, inhibitory or excitatory, will create graded potentials in that postsynaptic neuron, and that will determine whether or not an action potential will be generated in the next neuron. So then also the spinal cord, as you can guess, is a major highway for the upward and downward travel of both sensory and motor neurons. Sensory neurons going up, right, and motor neurons going down. So there are these major tracks, ascending and descending tracks. And again, the ascending tracks are of course going to be the sensory neurons that are bringing information to the central nervous system, or up the central nervous system, and usually up to the brain. And then the descending tracks are going to be bringing, again, information out to motor neurons. So when we look at a little bit of gross anatomy of the spinal cord, you're going to find that the spinal cord is going to be similar to the brain in that it's going to have the same meningeal layers, which we'll talk about in just a minute. But obviously the difference now is that it's protected by meninges, but now not protected by a skull, but of course the vertebral column. So again, the spinal cord is enclosed in the vertebral column, and if you remember, that vertebral foramen of each vertebra is where the spinal cord is going to run through. And the spinal cord begins at the foramen magnum, and then it ends, believe it or not, at about L1 or L2, usually somewhere in between those. And so this is kind of weird for most students to remember because the spinal cord actually ends well before the end of the vertebral column. And you'll see the reason for this is that spinal nerves, some of the spinal nerves will extend beyond the spinal cord, but the actual spinal cord itself is going to end at L1 or L2. Some of the major functions for the spinal cord, again, provide two-way communication to and from the brain. Major reflex centers, we kind of already talked about that in the last slide. So here you can see the spinal cord, and you can see there are all of these spinal nerves, right, that are entering, exiting. And I say entering, exiting because each spinal nerve will contain a mix of sensory and motor neurons. So some are coming in and some neurons are going out, carrying information out through that spinal nerve. So here are the spinal nerves that are entering, exiting the spinal cord, and then here's the spinal cord. You can see there are going to be two enlargements that we're going to talk about in the spinal cord. One called the cervical enlargement and one called the lumbar enlargement. And if I was in lecture, I would say, why is it that you think that we have these enlargements in these areas? What about the body would make sense that we would have enlargements in this area, in these two areas? And you can see, of course, that here, the upper limbs, okay, right around the cervical enlargement, spinal nerves will be innervating the upper limbs. So of course we're going to have an enlarged area in the spinal cord to accommodate all of that information coming in and out for the upper limbs. And then you guessed it, the lumbar region again is going to be for lower limbs. So that's why we have those two major enlargements. Here you can see right here, what we'll call just a minute, we'll talk about this in the next slide, the conus medullaris is going to be this little cone where the spinal cord ends at about L1 or L2. And then you'll see that spinal nerves will extend beyond the end of the spinal cord. So again, these are just nerves coming off of the spinal cord, and they'll extend downward or inferior below the spinal cord, and that's going to make up what's called the cauda equina. It's called cauda equina because it looks like a horse's tail. So caudal, again, is going to be tail, if you know the term caudal, and equina is horse. So this literally makes up the horse's tail, kind of looks like a horse's tail. So we'll talk more about that in just a minute, but that's kind of the basic kind of overview of what the spinal cord looks like. So again, just like the brain, the spinal cord is going to be protected by bone, except for this time it's vertebra instead of the brain, or instead of the brain, instead of the skull, right? And this time, then we have the same layers of meninges. Again, the pia mater is going to be directly covering the spinal cord, and then we're going to have the arachnoid layer, and then the dura mater on the outside there. And remember that just like in the brain, that subarachnoid space just below the arachnoid layer is going to carry the cerebral spinal fluid. And so of course then we have the cerebral spinal fluid that's going to protect the spinal cord. And again, the cerebral spinal fluid will run in that subarachnoid space around the spinal cord, but then it will also run through the central canal. So the epidural space, we'll have an epidural space above the dura mater, just like we did in the brain. And it's going to be a cushion of fat and a nice network of veins. And again, that'll be in the space between the vertebra and the actual dura mater. So if you look at the outer layer of the meninge, the dura mater, that space between there and the vertebra is going to be cushioned with that fat from the epidural space. So that'll provide some extra cushioning for the spinal cord in there and kind of help hold it in place. The cerebral spinal fluid, again, is going to fill up the subarachnoid space between the arachnoid layer and the pia mater. And you'll find, again, that that's going to also provide some cushioning, as well as, remember, the cerebral spinal fluid will bring in nutrients, ions, and remove wastes. And then when you look at the dural and the arachnoid membranes, they're going to extend to the sacrum beyond the end of the cord. And you'll see this, they're going to cover the phylum terminal, which we'll talk about in just a minute. And then L1, just to kind of talk about, but again, below that area of L1 is, or L2 to be kind of on the safe side, is a good site for a lumbar puncture. And let me kind of cruise past. The reason being is that, again, the spinal cord has ended. So if you're below L2, the spinal cord has ended. In between L4 and L5 is a good space because you're well below where the spinal cord has ended. And now, again, you're just really having to be careful of the spinal nerves. But this gives you a little bit more space to get into that subarachnoid space and draw out some cerebral spinal fluid. And it's a little less risky for you severing the spinal cord with a needle. So that's why they typically will do their lumbar punctures down in that area. And that's the nice part about having the meninge layers extend down, is that it provides a good area for us to be able to do lumbar punctures. So again, the meninges are just the same. The meninges are going to be the dura mater, the arachnoid layer, and then the pia mater right directly around the spinal cord. And again, the subarachnoid space, just like we talked about, is going to be the space where the cerebral spinal fluid will be. And that's, again, between the arachnoid mater and the pia mater. So again, hopefully this is all pretty familiar from lab, which is nice. This is actually a cross-section through a cadaver, which is kind of cool. So you can see some of the things things that we looked at in lab with that cross section. Here again, this would be a cervical vertebra. Here you can see the vertebral arteries, right, coming up through the transverse foramen. It kind of orients you. You'll see here's going to be your, and I'm just going to review this because we kind of talked about this. These are going to be your posterior and anterior roots coming in, and so those two together are going to make up the spinal nerve. So the posterior root would be running in the back here. This would be the anterior roots here. Here you can see it over here, anterior root, and then there's the posterior root, and the posterior root ganglion would be right down here, somewhere right here. And so anyway, it's kind of neat to see an actual cross-section of what we've been looking at. This is an actual cadaver, which is pretty neat. And that's the diagram of the lumbar puncture. So again, there are times when we need to remove cerebrospinal fluid. Sometimes they're looking for autoimmune diseases. Sometimes they're looking for bacteria to make sure that there's not a bacterial infection inside the cerebrospinal fluid. If there is a bacterial infection, it can cause inflammation of the meninges, and this is called meningitis. And obviously that's a much more serious condition because now you have bacteria again that have kind of breached that blood-brain barrier and are more likely to cross into the brain. So that's a much more serious scenario in terms of infection. So again, once we've breached the meninges, it doesn't guarantee that the bacteria will make it into the brain, but they have a more likely chance. So normally, again, an individual with meningitis is very sick, and it's taken very seriously, and they quickly put them on very, very high dose antibiotics. And again, they talk about doing the lumbar puncture. Again, the lumbar puncture needs to be safely below that second lumbar vertebra so that you're not risking hitting the spinal cord at all. So again, I believe nowadays between the 4th and 5th is more common for a lumbar puncture. The spinal cord again is the spinal cord begins as kind of a continuation of the medulla oblongata. So the medulla oblongata basically runs into and becomes the spinal cord, and terminates again at about the second lumbar vertebra in an adult. So below that L2, you're usually pretty safe to, again, have the spinal cord, the end of the spinal cord past that point. Again, you're usually safe to do a lumbar puncture below that, and again, that's why they usually pick the farther down you are. L4, L5 is a safe region. And we talked about the cervical and lumbar enlargements. Again, these are enlarged areas of the spinal cord because they serve as points of origin for nerves going to the extremities, the cervical being nerves going to the upper extremities, the arms, and then the lumbar nerves going to the lower extremity of the legs. So we'll look at the tapered portion of the spinal cord that I talked about. Is called the conus medullaris, and that's, you'll see, from which you'll see the filum terminale will arise and help to hold the spinal cord in place, and then the cauda equina, which are all the spinal nerves that go below that. So here are all of these things, and I'm going to use my pen here if I can without, you'll have to excuse me, it's been a little while since I've used my pen here. So this portion right here where the spinal cord ends, right here, this is called the conus medullaris. And you can see why it's called conus, because it's very cone-shaped, right? So that's the conus medullaris, and that's literally the end of the spinal cord. Now, to kind of anchor the spinal cord in place, and it's kind of hard to follow up here, but they have what's called the filum terminale. And the filum terminale is going to be just this kind of fibrous extension of the conus, and it'll be covered with pia mater, and it's going to help to anchor the spinal cord. So here, this is actually, and it'll run all the way up. And again, you can't see it very well in this because it's kind of hidden by the spinal nerves, but it's going to run all the way up here. So if you could picture oops, whoa, I got a little crazy with my pen there, sorry. If you could picture this running all the way down, this is the phylum terminal. And again, it's just going to anchor the spinal cord so that because it ends up at, in between L1 and L2, it ends so high up here. We have this phylum terminal extension that will actually anchor the spinal cord in place and hold it in place. And then the cauda equina, of course, is what's called the horse's tail. Literally, again, caudal means tail, equina means horse. So the cauda equina are all of these dorsal and ventral roots of the spinal nerves that are going to be running below the actual spinal cord. So all of this stuff down here, all these spinal nerves here are making up the cauda equina. So sorry, I said caudal because that's where the word comes from. So all of these spinal nerves here are making up the cauda equina. So, and again, the nice part is its name sounds just like what it is. Again, it's literally the horse's tail. So, and this was just again, a picture to show you that cervical enlargement right here, and then the lumbar enlargement down here. So you have that lumbar enlargement down there. Again, for the upper limbs, the cervical enlargement, and the lower limbs, the lumbar enlargement. These are just kind of some fun pictures you can look at these are actual cadavers, so you can see a little bit of the gross anatomy of the spinal cord. Excuse me here for a moment. So again, this is kind of so you can see the beginning of the spinal cord as it runs up here into the medulla oblongata. At the foramen magnum, and then you can see all of the spinal nerves coming off. This is kind of cool because they actually dissected this back so you can actually see the dura mater. So they cut open the dura mater so you can see that, and then you can see some of these spinal nerves again coming through. Here's the posterior dorsal root ganglion. And this is kind of nice because again you can see some of the structures. Here's the cauda or the conus medullaris. And then here, this right here is part of that extension called the phylum terminal. And then all the rest of this is all cauda equina. So when we look at our spinal nerves, we've got 31 pairs of spinal nerves, right? One coming at the left side, one coming at the right side. And they actually number them based on their cord level, coming in and out of the vertebra. So we have 8 cervical nerves, C1 through C8, because we have one pair. You may say, well, why do we have 8 and we only have 7 cervical vertebra? That's because one pair is going to come out above the first cervical vertebra. So that leaves us space for 8 cervical nerves. So everything else fits what you learned with the vertebra. You just have to remember that with the cervical nerves, you have one more cervical nerve than you do vertebra. So you've got 8 cervical nerves, 12 thoracic nerves, which fits normal, right? 5 pairs of lumbar nerves, and then 5 pairs of sacral nerves, 1 pair of coccygeal nerves. So again, remember, if you remember that the sacrum is 5 vertebrae fused together, it makes it easier to remember there are 5 pairs of sacral nerves. So all you have to remember is the same thing that you did before, except for, your 7, 12, and 5 when you eat breakfast, lunch, and dinner. If you like to eat breakfast a little late, because you like to get up a little late, 8 o'clock, right, is a better time for you. So 8 for cervical nerves there. And again, remember, most of these spinal nerves are mixed sensory and motor nerves. So again, you've got in each of these individual spinal nerves, you have, because a nerve is made up of, again, hundreds or even thousands of neurons coming in and out. Some of them are sensory neurons bringing information to the central nervous system, and some of them are motor neurons exiting the central nervous system and going to innervate a gland or a muscle. So then when we look at a closer look through the cross-section of a spinal nerve, this is kind of what we've already done in lab, which is nice. So if you look here, the spinal nerves begin as roots, and we have a dorsal or posterior root, And remember, the way that we can identify that dorsal or posterior root is by, of course, this lovely ganglion here. So we look for that ganglion, that bulge, where again we'd have a bunch of cell bodies. And the reason that we have that is because our sensory neurons are mostly unipolar neurons. So because they're mostly unipolar neurons, they have that weird structure where the cell body is just kind of hanging out on the side. And so as we have all of these sensors, they only show one here, but remember we've got, again, sometimes hundreds of sensory neurons coming in through this one area. We're going to have a bunch of cell bodies hanging out here. So that's what this ganglion is a big group of cell bodies from these unipolar sensory neurons. And so that's why the dorsal or the posterior root is going to have that ganglion. Now the anterior root is going to be mostly your motor neurons. And so those motor neurons are going to be multipolar neurons. And they don't have that weird structure where they have the cell body hanging out. Again, the cell body is part of, it kind of is continuous with the axon. So because of that, they don't have a ganglion hanging out here with the cell bodies. So again, that's kind of some of the basic breakdown of the anatomy that we looked at in lab. Also, when we do a cross-section of the spinal cord, we'll see that there are two lengthwise grooves that divide the spinal cord into the right and left halves. So on the anterior side, or ventral side, whichever one you like to use, we have that median fissure. It's actually, again, a little fissure, just like a crack in a rock. And then on the dorsal or posterior side, we just have a groove called the posterior or dorsal median sulcus. And then the gray matter, again, is going to be located in the core. The white matter is going to be on the outside. This is, again, opposite from what we think of with the brain with that outer gray matter cortex layer. And the central canal will run the length of the cord, carrying cerebral spinal fluid right through the middle of the cord. So here again you can see there is the central canal right in the center there, and then you can see here are your sensory neurons coming in here, and again we've got that dorsal root ganglion where all the cell bodies are going to hang out, and so that dorsal or posterior roots coming in here. Then here is the anterior root carrying, again, mostly motor neurons going out. So that's just kind of a review. Again, then this is kind of nice because it shows you we have all those same layers. We've got the epidural space right here with the fat. Then we have the dura mater. Then we have the arachnoid layer and the subarachnoid space where we're going to have, again, some cerebral spinal fluid in that subarachnoid space. And then the pia mater is this layer that's going to directly surround the spinal cord. So again, the pia mater is going to hug either the spinal cord or the brain, okay? Just as kind of a review. And so again, this is just another picture so that you can see the dorsal root ganglions just a little bit better. Now, when we look at the gray matter, again, this is hopefully mostly a review. Here's our gray matter in the center of the spinal cord, and we can break up our gray matter into the different horns. Again, the gray matter is going to be shaped like a big H, or I think it's shaped more like a butterfly. So you can see here's your butterfly shape here. And again, it's going to contain mostly cell bodies, unmyelinated axons, and dendrites. So that's why it's gray matter, right? The white matter is the myelinated axons. And if you look again, we're going to have these horns, these paired horns. So again, we'll have the up here. If you want to think of it to look for the anterior, if you don't have, like in this picture here, because we don't have the anterior and posterior roots of the spinal nerves. So the way that you can orient yourself is the large wings of the butterfly are going to be the more anterior region. So here's the anterior gray horn up here. Okay. Then we'll have the lateral gray horn, which would be right here. Okay. And then we'll have the posterior gray horn back here. So again, those are the horns. We have the dorsal. Again, I know that you probably are more likely to be using anterior and posterior, but again, I'll use both on the exam. whichever one you're most comfortable with. But posterior or dorsal gray horn is back here, lateral horn here, and anterior or ventral, if you like that, gray horn is here. Then remember that the gray commissures are going to be these neurons here that are going to be crossing the left and the right hemisphere. So this is the gray commissure. And again, we've got the anterior gray commissure on this side and the posterior gray commissure on this side. So again, those are just neurons that are connecting the right and the left hemisphere. And you'll see why in a minute when we do the reflex arcs, why this is important. And again, the central canal right in the center here is continuous with the fourth ventricle of the brain, where the cerebrospinal fluid, again, is made in the ventricles of the brain. That fourth ventricle then continues on into the central canal. So gray matter is going to be divided up into 4 groups based on somatic or visceral innervation. Remember, somatic is body, visceral, viscera, like I don't like the visceral mass, is going to be your organs. So we have, it's very easy, somatic sensory, visceral sensory, and then of course we have visceral motor and somatic motor. And what's cool is if you look at the spinal cord, the gray horn, the interneurons that are receiving some of that information, or again, you might have the cell bodies, whatever of those neurons that is making up the gray matter. They're broken up into sections. So you can see, and this is why, again, remember, it's easy to remember this because if you remember that here, the posterior root is going to have that posterior root ganglion. So that means we've got sensory neurons that are coming in with those weird unipolar neurons because those are sensory neurons. And they're going to come in. So, of course, it would be the posterior gray horn that's going to process the sensory information. So here we're going to have somatic sensory being innervated. Here we're going to have visceral sensory being innervated. So you can see the lateral and the anterior horn are going to be mostly dedicated to motor then. Because again, our motor neurons are exiting that anterior root. So here's our visceral motor and our somatic motor neurons. So when we look at the white column, you're going to see our white column is going to follow the same pattern in terms of posterior being mostly sensory, anterior and lateral being mostly motor. So remember that the white matter now is going to be our myelinated axons. That's why it's white, right? It's that myelin that makes it look white. And the white matter is going to be divided up into these columns. So again, the easy way to identify this for lab purposes, look for that posterior root ganglion. Boom, this is your posterior root, or your posterior column, sorry, your posterior white column. And so here's your posterior root, but there's your posterior white column right here. So then that tells you that this is the anterior white column, right? And then here are going to be the lateral white columns on the side. So again, each column is going to contain distinct bundles of nerve axons that have a kind of similar origin or common origin in each of these bundles. And so I'll show you this in just a minute. And we call these bundles tracks. They're literally like tracks of neurons. So you can think of them as these literally tracks, again, of either sensory neurons bringing information to the brain or motor neurons carrying information out. So again, we've got tracks, tracks are basically these highways for nerve conduction. And we're going to have ascending tracks that are bringing sensory information to the brain. So those are going to be more posterior, right? We're going to look for those more posterior. And the motor tracks that are conducting impulses down the cord and out to a muscle or a gland are going to be more anterior and lateral. So if you can excuse my maybe a 3rd grade level writing there. I'm still getting used to the pen again here. So the gray matter again receives and integrates both incoming and outgoing information, sensory and motor information. So you can see, again, just same trend, right? And this is the nice part, you get the same trend. So mostly, again, your posterior column is going to be sensory. All this posterior column is going to be sensory myelinated axons. Okay. You have a little bit of sensory on the periphery here of the lateral white column, a little tiny spot up here in the anterior. But for the most part, again, I want you to remember that sensory myelinated neurons, those tracts of sensory neurons, are mostly coming in the posterior column. So here that leaves our anterior column and our lateral column, white column. So our anterior white column and our lateral white column are mostly motor tracts, okay, for the most part. Again, we have a few sensory on the periphery, but the majority of the bulk of the lateral and the anterior white column are motor tracts, okay? So, and again, you can see that here. This is just another image of that. So just kind of some interesting things before we get into reflexes. Spinal cord trauma and disorders. If we have we can have localized injury to the spinal cord, or to the spinal cord roots, and that can lead to certain functional losses. So we'll talk about these different types of losses. If we have damage to the sensory tracts or the dorsal roots, right? And the dorsal roots, so where the dorsal root ganglion are what are carrying the sensory neurons. If we have damage in those areas, this is going to lead to what we call a sensory function loss. So you still have motor control, but you don't have all of the sensory information coming in. So we call this paresthesia. Okay, that's paresthesia. Now, for paralysis to occur, we have to have damage to either the ventral roots or the ventral horn cells. And so these, again, are going to be where we're carrying, remember, ventral or anterior, same thing. Our anterior ventral roots are going to be carrying our motor neurons. So this is where we can see actual paralysis. And paralysis is when we've done damage to the motor neurons. And this would lead to a loss of motor function. And there are two types of paralysis that we'll talk about real quick. There's flaccid or spastic paralysis. And both of them would be equally, I think, frustrating. So flaccid paralysis here is where we have severe damage to the ventral root. And so the impulses are blocked from this point where the damage is. And so impulses don't reach the muscles. So there's no voluntary or involuntary control of those muscles that once used to be innervated by those neurons that are now damaged. This will eventually lead to muscular atrophy because literally the muscles then aren't getting any impulses and the muscles just atrophy from disuse. So that's flaccid paralysis. Spastic paralysis is damage to some of the upper motor neurons inside the primary motor cortex of the brain. And so the spinal neurons actually remain intact, so there's not actual damage to the spinal neurons. It's actually the damage to the motor cortex in the brain, the primary motor cortex that's processing and helping us to, again, control those voluntary movements. And so the muscles are stimulated by reflex activity, but there's no voluntary control of the muscles. And oftentimes the muscles will start to shorten permanently because, again, they're just getting the reflex activity. So kind of interesting, that's called spastic paralysis. But again, that's because this is damage to the primary motor cortex in the brain. Flaccid paralysis is where we actually have damage to the spinal nerves, motor spinal nerves themselves. And then transection or cross-sectioning of the spinal cord at any level is going to result in, obviously that's our most severe trauma to the spinal cord, is literally the spinal cord is severed, right? At a point, then it can result in total motor and sensory loss in the regions that are below that cut because, again, there's no connection from the brain out to the periphery. And so paraplegic would be be an individual that has a transection somewhere between T1 and L1. I know that's a wide range, but depending upon what loss of function they have depends upon the type, how severe their paraplegia is, depending upon, again, where it is between T1 and L1. But again, that causes paraplegia. So again, more of that loss of motor and sensory information from the waist down. Where quadriplegia, we call it quad because you're going to be losing sensory and motor of all limbs. So again, that would be of your upper limbs and your lower limbs, and this is going to be transection somewhere in the cervical region. And then spinal shock is just some kind of transient, again, period of functional loss caudal to like a lesion. So again, this transient meaning kind of the in and out functional loss. That's caudal to if you have a lesion, if you haven't completely severed the spinal cord, you just have a lesion to the spinal cord. Poliomyelitis, I'm not going to get into this too much, it'd be fun to, but you can kind of read about it. This is just destruction of the ventral horn motor neurons by the actual poliovirus, depending upon, again, the damage to the motor neurons. If the motor neurons are completely damaged, the muscles are no longer getting innervation and muscles can atrophy. And death by polio, again, used to occur more from paralysis of the respiratory muscles, especially the diaphragm, because the motor neurons that innervated the diaphragm were damaged, and so the person could no longer breathe. Same, or the motor neurons that are innervating the heart were damaged, and their heart could no longer beat. So again, that's obviously why polio was something that we did a mass vaccination for, because polio could be so devastating, and it was such a severe virus. So with a nasty etiology. And then there's ALS, which I'm sure, the amyotrophic lateral sclerosis, which you've all probably heard of ALS, or Lou Gehrig's disease. And this is where we again see destruction of the ventral horn motor neurons and some of the fibers sometimes in the pyramidal tracts that you see again going up through the medulla oblongata. And the symptoms of this end up being a loss of ability to speak, swallow, and eventually a loss of the ability to breathe. Death, unfortunately, typically occurs within these individuals within 5 years. And they're still unsure as to all of the basically etiology, the causes of ALS. They know that it's epigenetic, so there's a genetic factor, but they also believe that there are environmental triggers, and so there's quite a bit of research obviously going into that. There are some treatments again for ALS, but with minimal success. Again, it's more of kind of an extending life type thing. Okay, so now we're going to get into some of our neural pathways and our major kind of spinal tracts, and eventually we'll get into our reflexes here. There are 4 things to keep in mind about the spinal tracts and pathways. One that we have, this decussation, right? And this mostly happens up in the medulla oblongata. So this mostly happens again in higher areas as you move up above the spinal cord. But most of your pathways, again, are going to cross from one side of the central nervous system to the other at some point. So that, again, motor control over, the right side of the body is really controlled by the left side of the brain and vice versa. So that's called decussation. Really, again, we're going to see that many of these neurons going up to the brain consist of a chain of at least 2 or 3 neurons. So we'll talk about these neurons and how they synapse on their way to the brain, and we'll talk about what are called first, second, and third order neurons in this relay to the brain. And then somatotopia just means that there are, if you can picture the homunculus, remember the homunculus that we talked about with like the sensory homunculus, where there are precise spatial relationships in the central nervous system that are going to correspond to certain areas of the body. So again, you can go look at certain areas in the central nervous system, especially in the brain, where you have certain parts of the brain that are going to be associated with the face. So in another part of the brain that's going to be associated with sensory information coming from the neck, and another part from the chest, and another part from the arms. So that's all that somatotopy is saying. You can just kind of picture that homunculus. And then symmetry is that we've got pathways that are paired. So we've got right and left pathways. And so that creates our symmetry of the spinal cord. So when we look at our ascending pathways, our sensory pathways on the way up towards the brain, we can see that there's this relay of neurons that I talked about. And there's usually, again, a chain of anywhere up to 3 neurons on our way up to the brain here. So, oops, get my pen working there a little better. So we've got what are called our first order, second order, and third order neurons. Our first order neuron is just like it sounds. It's going to be the first neuron in the relay towards the brain. So this is going to be the neuron that's going to be taking impulses from cutaneous receptors, meaning receptors at the skin, and then proprioreceptors in the muscles, right? So it's going to, first order neurons are taking information from those primary sensory areas in the skin, again, in the muscles as proprioreceptors, and carrying them back to the spinal cord or all the way up to the medulla. Okay, so they'll usually synapse with a second order neuron somewhere in the spinal cord or the medulla. So again, they're going to synapse with our second order neuron. Our second order neuron is going to be what's called an interneuron. So that interneuron, again, is going to be a neuron between the sensory neurons and other neurons in the brain or the spinal cord. And so the second order neurons are going to take that information and they're going to extend all the way up to the thalamus. So again, remember, they're going to synapse their first order neuron in the spinal cord or in the medulla. And then they're going to extend all the way up to the thalamus, remember, which is our relay station of the brain, right? Or they're going to go to the cerebellum, okay? And our second order neurons would end in the cerebellum. If this second order neuron goes to the thalamus, then it's very likely to synapse with the third order neuron in the relay. So again, the third order neuron is also an interneuron going between, again, another neuron in the central nervous system and a neuron in the brain. So, or a neuron here, in this case, from the spinal cord, or spinal cord, well, sorry, not spinal cord, so a neuron from the thalamus of the brain or the cerebellum to another higher region of the brain. So our third order neuron, again, is going from the thalamus on up into the somatosensory cortex. So third order neurons are going to only go from the thalamus to the somatosensory cortex. Sorry, I might have made that a little confusing, because if a second, like I first said, if a second order neuron ends in the cerebellum, then that's its final destination. Okay? If the second order neuron extends in the cerebellum, it's done. That's its final destination. It's carrying that information to the cerebellum. If the second order neuron ends in the thalamus, then it's going up into higher areas of the brain, especially the somatosensory cortex. So third order neuron is going to go from the thalamus to the somatosensory cortex of the brain. There are no third order neurons, again, in the cerebellum, because that's its final destination. So here you can see, I'm just going to kind of walk you through. Here's a first order neuron. So here you can see this first order neuron's a touch receptor on the foot, and it's going to come in through that dorsal root, right, or posterior root into the spinal cord, and then move up the spinal cord here, where eventually, again, it can synapse either in the spinal cord, or in this case, it's synapsing in the medulla. This is the medulla oblongata here. So it's synapsing in the medulla oblongata with a second second order neuron. Then the second order neuron is going to go from the medulla all the way up here into the thalamus of the brain, where then it will synapse with a third order neuron, and that third order neuron again will take it to the primary somatosensory cortex. So those are that's the three neurons in your sensory relay. Now, if it's going to the cerebellum, you're going to see you have this first order neuron, and it's going to come into the spinal cord, synapse here in the spinal cord with a second order neuron, and the second order neuron is going to go boom all the way to the cerebellum, and then it's done. Okay, so those are just the types of ascending or sensory tracts that you'll see coming into the brain. So now that we've kind of talked about some of these tracks, we can talk about some of these fun things like reflexes and reflex arcs. So a reflex, as you probably know, is a very fast and predictable automatic response to changes in the environment, especially to help us maintain homeostasis. Reflexes can be spinal, cranial, somatic, or autonomic, obviously, but we're going to focus on spinal reflexes because, again, they're the most common reflexes that we're going to see here, and we're doing the spinal lecture, so we're really going to just focus on the spinal reflexes. So again, reflexes can be inborn or intrinsic, as many of them are, but we also do have some learned or acquired reflexes as well. And they're going to involve only peripheral nerves and the spinal cord. So again, because we're going to be focusing on spinal reflexes. There are other reflexes that involve higher centers of the brain as well. We're going to kind of focus on these reflex arcs through the spinal cord. So here are the 5 components of our reflex arc, and you probably kind of already know these. So we have to have some type of sensory receptor that's responding to a stimulus, okay? And they'll send the information into the spinal cord through a sensory neuron. Then in our sensory neuron, we have to have an integrating neuron or an association neuron. So that's our integrating neuron. And then that'll synapse with some type of motor neuron that will travel all the way out to some type of an effector. And that effector, in this case, was a muscle, but it could also be a gland, okay? So effectors can be either muscles or glands. So literally, again, our motor neuron is either going to secrete neurotransmitters that are going to cause muscle contraction, or they're going to cause a gland to secrete something. So again, our reflex arc is kind of the simplest type of pathway. And typically, again, these are reflex arcs have the purpose of creating a very rapid response. So they're very simple pathways so that we can, create this rapid response for if you stick your hand on the hot stove. Obviously, you want to pull that hand away as quickly as possible. So we have these fast reflex arcs to protect the body. Again, helping maintaining homeostasis. So we're going to talk about some of these types of spinal reflexes. One that we're going to talk about is the stretch reflex, especially the patellar stretch reflex. We'll talk about the tendon reflex, which is kind of the opposite of the stretch reflex. And then we'll talk about the flexor withdrawal reflex and the crossed extensor reflex. So, and again, this is just showing you just what we talked about with the reflex arc. So you'll see that we've got different types of sensors. The Golgi tendon organs that we'll talk about in the Golgi tendon reflex are going to be mostly proprioceptors, kind of constantly informing the brain as to the state of the muscle. And so we'll talk about these. These are one of the types of sensors that we're going to see one of the reflexes, our Golgi tendon reflex with. We have another type of sensor in the muscles. So this is actually in the tendon. And again, it's mainly a proprioceptor, meaning that it's helping to tell the brain where that tendon is in response, in spatially where that tendon is from the muscles, okay? So if that tendon is tightening, that proprioceptor is detecting that change or that movement in that tendon from the rest of the body. So from the rest of the leg, okay, if it's a Golgi tendon like the patellar tendon, if that makes sense. And then stretch reflexes are going to be initiated by muscle spindles. We'll have these muscle spindles, and this will make more sense once I can show you a visual of this. But these muscle spindles are going to wrap around muscles, and they'll be initiated by the stretch of those muscles. So you can see here are these muscle spindles. These are a sensory receptor, and they literally are going to wrap or spiral around the muscle cells. And so they're going to detect stretch basically in the muscle cells. So again, if you stretch a muscle, then it'll detect that stretch and send an impulse. And then again, if the muscle is contracted, then it'll loosen up on that muscle spindle. So the muscle spindle, again, is firing when it's stretched because it's stretching, again, that sensor. So this first one is our stretch reflex. Actually, this one's probably, I think, from your book. And your stretch reflex is also called your patellar. This particular one is called your patellar stretch reflex or patellar tendon stretch reflex. And this one is going to be initiated by these muscle spindle receptors that are going to be surrounding, again, muscle fibers in the quadriceps muscle. Okay, so as your doctor pounds on your patellar tendon, right? Hopefully it's not pounding, but as he taps your patellar tendon, when it does that, it's going to stretch the quadricep muscles. And when it stretches those muscles, it's going to stretch those muscle spindles and stimulate an action potential. So that will travel all the way up this sensory neuron into, again, that dorsal root or posterior root into the spinal cord. And now it's going to synapse. And here in this case is more common with sensory neurons coming in. They'll branch and synapse oftentimes with multiple neurons, sometimes with an inner neuron, sometimes directly with a motor neuron. So here in this case, it's going to synapse on this side directly with a motor neuron, okay? And that motor neuron is going to travel all the way down to the quadricep muscles and cause the quadricep muscles to contract, kind of opposing that stretching of the muscle. And this is to help us maintain posture. The idea behind this reflex is that if for some reason your knee starts to buckle, we have this reflex that can help to initiate contraction of the quadricep muscles and help you maintain posture. So again, it's going to cause the contraction of the quadricep muscles. Now what's interesting is here you see the sensory neuron also branches with an interneuron that's going to branch with another motor neuron that's going to the opposing or antagonist muscle, the hamstring. And what's interesting is it inhibits. It's an inhibitory potential here. So again, this interneuron that's stimulated is inhibitory, and it will release inhibitory neurotransmitter, again, causing hyperpolarization in the cell body there and prevent this motor neuron from being excited, so to prevent muscle contraction of the hamstrings, which is the antagonist muscle. So again, kind of interesting. This is called our stretch reflex. So, and again, this is why when they tap on your knee, why your leg's going to jerk forward, again, causing the contraction of the quadricep muscles. Now, the Golgi tendon reflex is just the opposite. The Golgi tendon reflex is to prevent too much, basically, if a muscle is contracted, it will kind of prevent overstretching or damage to a muscle. So here you're going to see, in this case, you have a sensory neuron actually in the tendon, okay? And as you pull your leg back, you're going to see that it's going to stretch this tendon out, and or I should say, as you contract, actually it's the opposite. As you contract, it's gonna kind of pull taut on that tendon, and that's going to create an action potential in your sensory neuron. That sensory neuron will come here, and again, it's going to branch and synapse, and it'll synapse with neuron here that will inhibit actually the quadricep muscles. So it'll inhibit the quadricep muscles and now stimulate. So this is just the opposite of the reflex that we just talked about, and stimulate your antagonist muscles to contract, which would be the hamstring muscles to contract. So pulling your leg back. And so this is, again, just to prevent basically damage to the muscle from and tendon as it's contracted to make it too taut, if that makes any sense there. Let's see if there's anything else I was going to tell you about that. So yeah, so this reflex, again, mainly is to help prevent the tearing of muscles and tendons. It's kind of again, this is not the more, Golgi tendon is not the kind of more common talked about reflex. And this might be kind of a better image of it here, because you can see the blue in here. It shows you again that there's the sensory neuron coming in, and then you can see the inhibitory in red, inhibiting the quadriceps, and here in purple, the hamstrings are being simulated. So you can think of this as being your kind of flexor reflex, and this is your extensor reflex. So causing, again, your leg to extend. This is your flexor reflex, causing your leg to flex. So that's probably kind of the easiest way to consider these reflexes. Here, there you've got your extensor stretch reflex and your flexor Golgi tendon reflex. If that'll probably make it a little bit easier in terms of the explanation of that. And then this one's really cool. This is called the crossed extensor reflex. And this one is one of my favorite reflexes to talk about, because it's just a cool reflex. It makes sense, right? If you were to step on a tack, you, want to flex, right? The leg that you just stepped on the tack with, to pull it away from the tack. But if you're going to do that, you want to extend the other leg, right? To kind of help hold you in place. Otherwise, you're going to buckle and fall over. So this is kind of a cool reflex. You can see here, and that's why it's called the crossed extensor reflex, because the leg that you step on the tack with is going to stimulate neurons on the other side, motor neurons on the other side of the spinal cord, and cause extension. So here you can see you step on the tack with your right leg, and that sensory information is going to come to the spinal cord. You're going to, again, meet up with synapse with a second neuron here called your association neuron, or your interneuron here. Association neurons or interneurons are the same thing. So here's your interneuron here, and then that interneuron is going to synapse with a motor neuron here. That's going to contract or basically stimulate and contract your flexor muscles, which are your hamstrings. So again, that'll cause you to withdraw your right leg. Now, the cool part is that you also are going to synapse with these neurons that are going to cross, these interneurons that are going to cross the gray commissure into the left side of the spinal cord, and now synapse with motor neurons that are going to control the quadriceps, the extensor muscles, right, of the opposite leg, the left leg, now causing that leg to extend so that it can help to stabilize you and keep you standing, right, basically, so that you're not buckling when you step on a tack with the opposite foot. So anyway, this is just kind of a fun reflex to talk about. It's called the crossed extensor reflex. And again, pain signals will cross the spinal cord to the opposite side through that gray commissure and cause contralateral extension. Contralateral meaning the opposite side, right? Because we stepped on the tack with the right, we're causing extension with the left. That's contralateral extensor muscles to be stimulated. So again, pretty cool. And that way then you can, again, have that left leg will maintain body weight. while you painfully now go pull the tack out of your foot. Hopefully none of you have experienced that firsthand. You can see they're just showing you cross extensor reflex with the arms, but I think it's kind of a neater one to show with the legs, so I'm using the tora one here for the legs. There are some other superficial reflexes that are fun to talk about that are more reflexes really to kind of look at the functioning of the nervous system. So you can talk about the Babinski reflex. The Babinski sign is kind of an abnormal plantar reflex. A normal plantar reflex is initiated if you stimulate the lateral aspect of the sole of the foot. The response should be the downward flexion of the toes, meaning your toes will kind of curl down. If you look at an infant that still has the Babinski reflex because we haven't had the development of the nervous system and especially the myelination that's needed on the neurons for proper functioning of these reflexes. So pre-myelination of those neurons, you can see the Babinski reflex. And instead, they'll get an abnormal plantar reflex where they'll actually take their big toe and dorsiflex or pull back their big toe, and then the smaller toes will fan laterally instead of curling under. So that's called the Babinski sign. It's kind of fun to watch this on your infant as that Babinski sign slowly kind of goes away, and then eventually they have the normal plantar reflex. But obviously this would be a sign if for some reason you're an adult or even a child past the infant phase and you have a Babinski reflex, and this is a sign of damage, indicating what we call corticospinal damage within the spinal cord. Within the spinal cord or the cortex. So then we have these different connective tissue coverings. And so with our reflexes, guys, again, I just want you to be able to recognize the basic reflexes. Again, the basic reflexes of our, especially our extensor stretch reflex, which is the patellar stretch reflex. And then our Golgi tendon, which is our Golgi tendon reflex, is the flexor reflex. And then the crossed extensor reflex. Again, Babinski is just kind of a fun one to talk about. Again, you shouldn't have that Babinski reflex except for, again, in young infants. Most of us normally, if we have, again, a fully functioning nervous system, especially no damage to the spinal cord or the cortex, then we have a normal plantar reflex. So that's basically kind of what I want you to know with those. Again, just kind of more of the superficial of those. And then this is pretty easy because we did this with the muscles and we've kind of already done this in lab. So when we look at our nerves, we can see that the nerves, again, remember, are bundles of axons, right? And so here, they're going to branch that just like we took a muscle apart. Here, we can take apart a nerve and break it down into our fascicles and then our individual axons. So each individual axon is going to be surrounded by some connective tissue called the endoneurium, right? The endoneurium is going to surround each of these individual axons. Then these axons will be grouped together in fascicles, just like muscle fascicles. So here's a nerve fascicle. And the connective tissue that surrounds these nerve fascicles is, of course, called the perineurium, right? And then the connective tissue that's going to surround and hold together all of these fascicles is called the epineurium. The epineurium will go around the entire nerve. So this is nice because it's just like the muscle, except for instead of endomysium, perimysium, and epimysium, you have endoneurium, perineurium, and epineurium. And then the dura mater will kind of blend into this outer layer of the epineurium. So the dura mater is going to be the dura mater will again kind of cover that outer layer of each of these nerves. So we have dura mater that's going to cover the spinal nerves that will extend onto the spinal nerves, as they exited the spinal cord. So the dura mater kind of blends into that at the intervertebral foramen. So again, the dura mater just blends in at that intervertebral foramen, and then after that, the dura mater kind of just basically kind of blends in and then goes away into that epineurium. So let's see, and then this last one is going to show you more branching of the spinal nerve as it exits that intervertebral foramen. So again, kind of where our dura mater blends into that outer layer of epineurium and then just kind of subsides there. So here is our intervertebral foramen. And so we've really only talked about what's happened on the other side of the intervertebral foramen, where it branches here into the posterior root and the anterior root. But remember, they come back together here and form what we call the spinal nerve. And then it's going to branch again. I know, lots of branching, right? And when it branches on the other side of the intervertebral foramen here, we now call them, instead of roots, we call them rami. So this here will be the anterior ramus because it's anterior, and here is going to be the posterior ramus. And that's just branching oftentimes of the nerves going, again, the anterior ramus going more anterior and the posterior ramus going more posterior. And then you have this branch coming off of here that goes to the sympathetic trunk. And this, of course, is called the rami communicantes or ramus communicantes. Each individual one's called the ramus communicantes. And so this is where the spinal nerve is connecting or communicating with the sympathetic nervous system. And here's your sympathetic ganglion. Remember, the sympathetic ganglion will run all alongside here. You can see this, the sympathetic ganglion running all along this spinal cord here on either side, forming the sympathetic trunk. Okay. So the ramus communicantes are just a way to communicate with that sympathetic trunk because, again, the sympathetic nervous system is so tightly controlled, and we'll talk about this in terms of, you know, the sympathetic nervous system is going to help kind of control the motor neurons that are going for our fight or flight type activities. So, and we'll talk about those fight or flight activities when we get to the autonomic nervous system. So again, the dorsal rami is going to supply the skin and muscles of the back. Dorsal again, or posterior ramus is going to supply skin and muscles of the back. The anterior ramus is going to form the plexus that supplies the anterior trunk and the limbs. And we are not going to talk about them so much. We do have meningeal branch that will supply the meninges, the vertebra, and blood vessels. But again, that's kind of getting into some of the nitty-gritties a little more than we need to talk about. And then they don't have on here the ramus communicantes, which is more important that you know than really, for me, more than the meningeal branches, because we don't talk about the meningeal branches so much in lab. But we do talk about the ramus communicantes, and those are these branches here that are communicating with the sympathetic trunk. And again, this will make more sense when we get into the autonomic nervous system, but important to remember that for when we get there. So that is the end of this lecture. So I'm going to go ahead and stop this lecture. I'll post this one separately from the 45 minutes of the peripheral nervous system. Please, please, please feel free to email me if you have any questions or concerns, and I'll go ahead and I'll be posting the objective questions for this shortly as well.